As we continue on our discussion with cell growth and development, we're now going to shift all attention toward DNA. This is going to be important when we talk about how DNA is copied later on and kind of make a connection with the cell cycle in terms of what's happening during interphase. But before we get there, we have to learn about what makes up DNA. So if we talk about DNA, we first have to mention that we are dealing with nucleic acid. If you recall, a nucleic acid's building block is a monomer. Now we'll get to that here in a second. Now, we only have two types of nucleic acids. You have DNA and you have RNA. RNA is going to be more deeply discussed in our third part of Unit 5. But for now, we're only going to focus on DNA and really see what the differences are between the two. When we look at DNA and RNA, we're going to focus on three major differences. First off, this has two strands, and this only has one strand. That's why DNA is called a twisted ladder. There's two strands. The name itself, deoxyribo and ribo, they are named after their sugars. The sugar for DNA is deoxyribose, and the sugar for RNA is ribose. And then finally, they differ on a specific nitrogen base. Notice how both of them have cytosine. They both have guanine, so C still bonds with G. They both have adenine. But there is a difference now in what bonds with adenine. DNA has T, RNA has U. So our third and final difference is RNA has uracil and DNA has thymine. Now looking at specifically DNA, we have our nucleotide. This is our phosphate. This is our sugar, deoxyribose. And here is our base. In this case, we have C. The backbone of DNA is made up of alternating sugar and phosphates. And then the inside of the ladder, the rungs, when you think of a ladder, kind of where you step your foot on, that is going to be your bonded nitrogen base pairs. When we look at our DNA structure, notice how it looks like this is pointed up and this is pointed down. That's because the structure of DNA is what's called anti-parallel, which means that the two strands, strand 1 and strand 2, they run in opposite directions. And we'll go into much more detail about what that means later on. So we have our ladder, and then we have our double helix shape that is more realistic and more accurate in describing the shape. So the overall shape of DNA, Watson and Crick found with the help of Wilkins and Rosalind Franklin that it is a double helix. We have 4 bases that are part of DNA. And those bases are grouped into 2 groups. You have what's called your pyrimidines. And these are going to have a single ring. They're going to be a little bit smaller. And this is going to be T and C. And then you also have what's called your purines. And they have a double ring. And this is going to be A and G. Now the thing to notice here is a big one kind of bonds with a little one. So if we go back, notice how T and C, your pyrimidines. So here's a pyrimidine, T and C. They have a single ring. So they're a little bit smaller. Whereas your A and G are your purines. Double rings. So 2 rings. 2 rings versus 1. A big one, the double ring, pairs up with a small one. Now the backbone of your DNA, you are going to have phosphate group, so phosphate, and then you are going to have your sugar. And your sugar, remember for DNA, that's where it gets its name, is deoxyribose. So sugar, phosphate, backbone. So here's our phosphate right here, and then here is our deoxyribose right there. Our base pairing rules. T bonds with A. C bonds with G. Now, Chargaff was a scientist who came up with the idea that the amount of A equals the amount of T, and the amount of C equals the amount of G. Why this is important? Because in the discovery of DNA, these rules led to the idea that, oh, instead of them pairing up with each other, they must pair up with what they have equal concentrations with instead. So that's when we get your purines pairing up with your pyrimidines. We have 2 types of bonds. Within a DNA structure, you have your hydrogen bonds, and these are going to be between your nitrogen bases. And then you also have covalent bonds, and these are going to be between your sugar and phosphates. And remember, anti-parallel, from what we talked about in the slide previous, is DNA runs in opposite directions. So essentially, when we look at our 2 strands, one strand looks like it's going one direction, the other strand looks like it is going the opposite direction. So what I mean by that, when we look at our diagram here, with anti-parallel, we would say that this runs in a 5 to 3 direction. What I mean by that is if you count your carbons that is in the nucleotide, at the point we have an oxygen, and then you count your carbons from your oxygen in a clockwise fashion. So 1, 2, 3, 4, and then a 5th one's right here. Notice how our 5 to 3, and that's where we get that 5 to 3 direction. 5, where the 5 carbon is at the top, that position, the 3 position carbon at the bottom, 5 to 3 direction. And we see that with all of your nucleotides going the whole way down, 5 to 3, when your oxygens are pointing up. When your oxygens are pointing down, anti-parallel tells us that we're operating in the opposite direction. So we are going in the 3 to 5 direction. If we do the same count, and we go from the O, 1, 2, 3, 4, 5, going clockwise. 3 is at the top, 5 is at the bottom, and that's why we are going 3 to 5. And notice how it looks like it's a little, upside down compared to what the other strand was. That's anti-parallel. One strand going one direction, the other strand going the other direction, and understanding 5 to 3 or 3 to 5. That'll be important when we talk about replication. The final two things we're going to focus on, these bonds right here, those are your hydrogen bonds between your nitrogen bases, and then your bond between your sugar and your phosphate, deoxyribose and your phosphate. This is going to be your covalent bond. Last thing, we'll just kind of review how we get that 3 to 5 designation. You have your phosphate, and we essentially have something that looks more or less like this. I'm just going to say this is a G, that's our, or a C, or a G, whichever. That is our nitrogen base. Remember, this is your phosphate, and this is deoxyribose. The three things to make up a nucleotide, the monomer of a nucleic acid. Now, starting at your oxygen, go clockwise. We have 1, so kind of count your points. 2, 3, 4, 5. Since 5 is at the top and 3 is at the bottom, whenever we pair up more nucleotides to this, we would say that this is going in the 5 to 3 direction. That little dash stands for prime. That's simply identifying a position. 5 is at the top, 3 is at the bottom. So 5 to 3 direction. Okay, that is it for the structure of DNA. Let me know if you have any questions. Thanks for watching.